breakout question times.
Okay. So just so Solomon knows, so he can have those kind of ready.
And can let's see, once they come back, can I share my screen? I know sometimes that it's blocked.
No, you're good. You're set up as a co-host,
yeah.
so you should be able to share it, no problem.
Yeah, so we'll go through some slides and do the same thing.
Solomon, do you want to send Michelle, Santiago, and Abby into different groups and then see how, check on the students? Since it's kind of only one question, it's a long question, it probably won't take them quite as long this time.
Probably not.
Oh, they're already gone.
All right.
Okay. So Solomon, right now, are we the only thing streaming on YouTube?
Yeah. Yeah, okay. I don't know. I think this one probably would have been better to.
Yeah, probably just invite people in. Yeah.
I don't know how many people would have shown up, but imagine some. I guess the issue is if there's too many, because then it starts to mess with the activity.
Right.
All right, and continue going to the mutants.
Why don't you send a message to them and give them like a 3-minute warning?
All right. And then who did we want to double assign things to?
Maybe Trayvon and Amira.
All right. And assign them to the same one, or assign them each to one that someone else is already doing?
To one that everyone else is already doing. And then everyone else, it doesn't matter other than those. Or maybe actually Jenna as well, since she might be missing. We're just trying to get everyone covered.
Right.
Or as many as possible.
David, I just wanted to mention, unfortunately, I have another meeting that came up at 3:30.
No problem.
um,
No problem. I understand.
things, I'll tell you.
Yeah.
But everything going okay, generally?
Everything, yeah, generally, I think everything's going okay. Again, I think the important part of our program is the students and their research and their mentors, and everything seems to be going well there.
Yeah, it sounds like you've had, I looked at the Google Classroom, some really good speakers. Dr. Mendez.
Yeah, do you know her?
I do. I've met her before. Yeah, she's excellent. I'd like to get involved more with her, but I know she's so busy. So I'm glad she came to Hillman anyway.
Yeah, she gave a really excellent talk. So she was invited by one of our old students, Taylor Robinson, who went to Ellis, but has been part of our program for years. So she invited Dr. Mendez. She's at this graduate school of public health now, right. and the panel for public health too, which were both really excellent talks. So that was good. There we go. Hey, Jillian and Erica, how are you? Oh, and Sam.
Hi, Dr. Boone. I'm fine. How about yourself?
Doing well. Doing well. Were you guys group 1, 2, 3?
3.
3. Okay.
They're going to be called group 3. They were technically the second group. The way that makes more sense to break these out is to make an extra group that isn't going to be assigned and then just delete it for all the unused names.
Okay, I see.
So it doesn't let you like separate out co-hosts otherwise.
Do you want to close out the rooms now, Solomon?
Sure.
Try to get everyone back here. Here, everyone's coming. All right, I think we are all back. So Solomon, who was in what would be group 1?
That was Amanda, Angelina, Anissa, and Oscar.
So do one of you want to share your answers with the rest of the group? Who would like to volunteer? Amanda, Anissa?
Well, I talked a little about it with Dr. Matela in our individual groups. So maybe someone else from my group wants to talk about it. Or I could.
Go ahead, Amanda, that's fine.
Well, we were just talking about how like there are a couple different ways that like bacteria could either like segregate with their respective mutations, or like mutations could be similar even though they're completely like different populations. Like these mutations that are happening, all it's doing is to help them adapt to a new environment. So like whatever mutation would help the bacteria survive is what is going to be passed down through generations. And in the context of this question, like the mutations were different among the two groups of bacteria.
Did any other groups have a different response to that question?
I suggested magic.
I don't think, probably not magic.
But it could be.
Well, in science, we can deal with real phenomena. I think so. I thought that was a really good answer. You know, thinking about mutations are going to occur randomly. So perhaps in the different populations, you know, bacteria found a different way to be able to better attach to the bead. All right. So, okay, I'm gonna go through the next part of the slideshow. This is thinking about whether these changes we see to colony morphology, are they heritable changes, right? Can they be passed on to their offspring? So how would we test for that? You know, what do we think would happen if we inoculated, we picked one of these wrinkly colonies and then inoculated a new culture overnight and then plated a sample of those bacteria from the new overnight culture. We've done that. And really the answer is that all the colonies would be wrinkly because we know that these changes we see to colony morphology are a result of changes to the DNA, right? So those changes are going to be inherited by the bacteria's offspring. So thinking about, you know, we can look at changes in the population by seeing changes to their colony morphologies, but how could we actually test whether those new morphologies make those bacteria better biofilm producers than the ancestor? And one way we can do this, and this has been done at SciTech, is we can assay biofilm production using this stain called crystal violet. So crystal violet will attach to the biofilm. So we'll grow cells overnight, right? Pick the colonies, like a smooth colony or a wrinkly colony. Grow them overnight. In this case, we're growing them in these tiny little wells of a 96 well plate. And then we'll use, so the bacteria will grow and some of them will attach to the sides, just like you could see on the bigger test tubes. And then we can stain what's attached with this crystal violet dye, wash away the dye that didn't stick to the biofilm, and then we can measure how much dye is left over. So in this case, you can see these smooth ancestral colony is not very darkly stained, right? So not that much biofilm produced, but when we do that with a wrinkly colony, we can see that there's a way higher, you know, the purple stain is much darker, meaning that they were able to produce a lot more biofilm. And this is just another way that we can do this. So rather than using a 96 well plate, we can stain directly in the culture tube. So here's two different types of mutants. So one of the big spreaders and one of the ones that are those little wrinkly clumps. And here we're just adding the stain and then washing away excess dye. And if you look kind of closely, you can see that on the side, right, there's this big layer of biofilm there. And there's also a tinier layer, but a layer of biofilm that's stained in this test tube. And then if we add just alcohol just to dissolve the dye in the biofilm, then you can see that both of them have some amount of biofilm production. This one, from this kind of colony, much more than the smaller one in this case. And then you could actually, you know, go a little further to compare what the absorbance. So you can measure a reading of how much dye was produced, and then use Excel, for example, to plot that. And so you can compare how much biofilm was produced by the ancestor compared to different evolved colony morphologies. And we can see here again, so these are doing replicates within a plate. So that you can see it's pretty variable, the staining protocol. But in general, whenever we have these kind of wrinkly mutants appear, they produce more biofilm than the ancestor. Oh, so here's the next question.
Abby, before we break off into the groups to discuss these, Sam asked a question. It's about competition, I think. He asked, do bacteria fight one another or will they just ignore other colonies? So when there are more than one type of colony on a plate.
Well, the colonies won't interact with each other on the plates. So when you're plating the bacteria, you're trying to spread them out so that the colonies are formed from an individual bacterium that's reproduced multiple times to make its own little group. And so that group everyone in that group should be genetically identical in the colony. So they're not competing there.
So he asked, what if they do grow into each other? So if you were to co-culture them and they grew into each other, do they fight? Do they compete for resources? Or do they just ignore each other?
Well, I think depending on the genotype of the bacteria, they could potentially fight and compete for resources. And that would be a good thing to look at, right? Like put them close together and see if one kind of pushes the other one away. Yeah, that's a kind of a question you can test, but we've not looked at that. I think typically when if they're ever plated close together, it doesn't seem like one is pushing out a different type of colony with a different phenotype.
I have a question about that. So on the TEDx video, like it showed like different types of bacteria like living within one biofilm. So like I'm guessing this is different, right?
I think in some ways it's the same, but you're right. So I think in nature it's probably very common for bacteria to form multi-species communities, right? In our case, we're just looking at one particular species and how it comes together. But it also could be that they're competing with each other, those different species in the biofilms in the real world.
Any other questions before we break off? So why don't we look at the questions first before we break off? So,
I'll just, I'll add them to that little side.
Into the chat?
Yeah.
So the first one was asking about the crystal violet assay. Does anyone have a question about the crystal violet assay, what it's measuring, before we break off and ask you to think of an experiment on how to use it, since it was briefly covered here? No? Okay. So the first question is asking you to use, can you think of a research question that could be tested with the crystal violet assay? And then the second is, what would happen if you inoculated a new culture with a wrinkly and smooth colony and grew the bacteria for a week by transferring daily without the bead? And I think we'll break you off into groups again. If I'm doing this right, Abby, as I'm just trying to help, and maybe I should just stop talking.
No.
But I think we're breaking you off into groups for you guys to discuss this. And maybe when we come back this time, group 2 will help lead us through that first question to see if they can come up with a research question. And group 3, you can help us walk through what you think will happen if we inoculate these 2 colonies and don't transfer beads. And we'll give you a couple of minutes to, just like last time, to talk among yourselves. And then we will have people jump in the group with you in case you have any clarifying questions. Okay.
All right. Are you ready?
Yep. All right.
All right, and I finished assigning them to their mutants.
Okay, great. So Abby, whenever you tell them about their assignment, you can tell them that, let's see if I can find where it is. Where did you type it in at? Tell them.
If you go to the assignment overview README, I added like a little sheet, and then under each of the mutants themselves, I added a comment in the description as to who was assigned to it.
Oh, okay, perfect. Let's see if I can see this. Yeah, so if you click on each individual mutant, it has names there. Perfect.
Yeah.
So you can tell them that, Abby.
Okay. Oh, yeah, I see that. Okay. I think we should. Go ahead.
I just wanted to ask, crystal violet is biofilm production, right?
It stains. It's a stain.
It stains
for biofilm, so attaches to the biofilm. So darker color, more biofilm.
I got confused with concentration.
Crystal violet basically binds to DNA. So the more of the organism that's there, the more. We use it in mammalian cell culture as well.
Oh.
I think it attaches to lots of stuff, right?
Yeah, exactly.
Because I don't know if it's just attaching to the DNA in the.
It might not be in the bacteria. That's what we use it for. But it's probably anything that's negatively charged it's binding to. So maybe a gram-positive bacteria will just bind to the cell wall or whatever on the outside too. I don't know. I know nothing about microbiology, or very little.
All I know is that in organic chemistry, we did an experiment and then we tested the success of our experiment using crystal violet. And all of my samples turned purple because my experiment didn't go really well. So
it's a neat assay to be able to do. If people, we've thought about that. It takes a little longer. It's like a few more days, but and it doesn't work that well because this is just a experimental side. Typically, I think it doesn't work that well because actually, if the bacteria are really high biofilm producers, they'll make a mat. And so when you try to wash away the stain, you wash away the mat. And so they'll look like they have less. That's science for you.
Mm-hmm.
It's colorful though.
So bacteria cell walls are stained by crystal violet, just like DNA. See that? I should have been a microbiologist.
I wonder though, I even think in this case, it must stain whatever, the biofilm matrix, they're different for each cell, but they're like polysaccharides and it must be staining stuff in the matrix too. I know it's polysaccharides, proteins, and DNA, I think, depending on species.
All right, Solomon, do you want to send Abby, Michelle, and Santiago into their groups to see how everyone's doing? Did people talk to you guys last time? Or were they talking when you jumped in?
Mm-hmm.
Good.
Do you want to be in the same groups as last time or slightly different?
I didn't hear what you said, Santiago.
Oh, I just said my group wasn't talking when I came in.
Okay, so that's good. That's why we need to send you in.
My group made a Princess Bride reference and then they had to explain it to me.
All right, so put everyone in the same groups?
That sounds good.
Okay.
Same groups are fine.
There they all go.
All right. We can just give everyone a couple of minutes, give them like 2 minutes and then give them a 2-minute warning and then we'll bring them back.
All right.
Bring them back at 3:35.
And then is there another breakout session after this?
I think there's one more, is what Abby was saying. Again, Abby's kind of running the whole show. I don't really know what's coming, but I think there's one more.
So tired this afternoon. I don't know why.
It's Zoom. I'm telling
Yeah.
you. I think Zoom has
Zoom
made
is
me
very
age.
draining.
It is. I wonder if it's Zoom or if it's like you just need to get up and walk between places, like walking between meetings and things that
Yeah.
gives you energy or something. when you're just hitting end meeting and then clicking on another link to start a new meeting and still sitting in the same place.
Mm-hmm.
Yeah. Maybe I should do that. Maybe after every meeting, I should make myself walk like around my block before I come back and start a new one.
Yeah, that's what I try to do sometimes is not even just between meetings, but like if I'm just feeling myself like starting to fade or not really focus as well, I'll just take a second, like step up, walk around, stretch, and then get back in.
Yeah, I think it's a good idea.
Yeah, I wonder if like teaching curriculums will start to teach how like classes on how to better teach online classes.
I would imagine that that's already starting to happen. So why don't you give them a 2-minute warning?
All right.
We have one more breakout session. Yeah, that way I think we should have time to do everything. I don't remember it. Can people in the breakout rooms still see the same chat feature, or does it become new whenever you go into it?
So like the breakout, like if they had a chat from a previous breakout?
Yeah, like the chat from this main breakout, can the people in the breakout rooms actually see it? Or
I don't know if they can see it in the breakout room. I know if they come back, they can see it.
Yeah, I know that as well. I just don't know if they can actually see it.
Maybe we should just quickly ask whoever comes out first.
Yeah, we can do that. Or we can just jump into a breakout room real quick and see if their chat, still see their chat. Sure. Although, send me into some place.
All right.
All right. So Solomon, why don't you bring everyone back?
All right.
I could still see the chat though.
Okay.
So that's good.
Good news.
Yeah.
Welcome back, group 3. Now we got group 2. And I'm just waiting on group 1. Now we have group 2.
I think we're all back, Abby.
Great. And then, so which group, I can't remember, is it group 2 was gonna let us?
So this is gonna be group, the group with Erica, Julian, Marcus, and Sam.
So do any of you want to share your research question that could be tested with Crystal Violet, or what you think might be able to be tested with Crystal Violet?
You can test to see if as the bacteria evolve, if they're able to better produce biofilms. You can do that by either comparing the bacteria overall from day 2 or day 5, or just comparing the different colony morphologies on day 5.
Yeah, so you could say, you could wait till the last day of the experiment, and if you had a mixture of colony morphology, so you could say, well, how does that relate to their ability to make biofilm? So you could compare smooths to different kinds of wrinklies. And you're exactly right where you could go, in this experimental setup, not only can you do that, but you actually have a plate of bacteria or several plates that contain the ancestors of these later populations. So you could compare, you might even just compare smooth colonies, right? And say, so what is the biofilm capability of a smooth colony from the beginning of the experiment to a smooth colony at the end of the experiment? Like, even though they look the same, maybe the later bacteria are now better able to produce biofilm. They have more biofilm capability than their ancestors did because they're also being selected for that in this environment. Does anyone have any questions about that or had any other thoughts?
We were wondering if you could try and find out whether younger colonies or younger generations were always better at producing biofilm than older generations, or the ancestor generation, or if it ever got worse or stayed the same.
Let me see if I can. So you're wondering if the ancestral populations are ever better biofilm producers than the evolved populations?
Yeah, if that's possible, or if it's just always that biofilming improves.
Well, so I guess the answer isn't completely straightforward. Typically, the evolved populations are going to contain bacteria that are higher biofilm producers because that's what we're selecting for in the experiment. But it could be that some portion of the evolved population is equivalent to the ancestor making biofilm. Or, it really is it's just a testable hypothesis that, you could test. Could that happen? We take ancestor and compare it to the evolved and see, maybe biofilm production in some of them for some reason is actually reduced over time. Maybe because they're living in a biofilm of a high biofilm former, right? So there could be reasons why you might believe that they would actually make less biofilm at the end of the experiment.
Good question. And Marcus, great answer earlier. And so why don't we move on to the next question? And this would be group 3. So what do you think would happen if you inoculated a new culture with wrinkly and smooth colonies and then grew the bacteria for a week by transferring daily without the bead? So what did you guys think? We discussed this. And who was in this group?
This was Alexandria, Eduardo, Jonathan, and Jonathan.
You guys have any thoughts?
Maybe it would be mostly wrinkly colonies because weren't they better at producing biofilm?
So, and I think that in my group, we talked about this too. I think maybe the question wasn't completely clear, so I'll tell you how I was imagining it. So in the original selection experiment, you were using the bead to select for high biofilm colonies, which then look wrinkly, right? So if you take the high biofilm bacteria and compete them against bacteria that are lower biofilm producers, and then you're putting them back into their original environment, just they're free swimming, they don't have that bead anymore, right? So I would imagine if you select for that, likely producing biofilm is costly, right? And so they would probably be, high biofilm forming bacteria would probably be slower at reproducing. And actually those smooth ones would outcompete them because now they're in an environment where it's better to just reproduce really quickly rather than being selected to attach and stick to a bead. But of course, that's another great way that you can think about how you can extend the initial experiment to ask these questions and then see what happens.
Anyone else have any thoughts about this? All right. If not, Abby, you want to keep going?
Yeah, so we'll do, I think this is kind of the end. So now we'll just talk a little bit about, how are these studies relevant to medical applications? So in brief, biofilms are a major cause of infections, all kinds of different infections. I think someone had asked a question in the assignment about what's device associated mean. Device associated is just medical devices. So you could think of like catheters or ventilators, right, where bacteria might attach and reproduce there. Biofilm associated infections can be very costly, and one reason that biofilm infections are so detrimental is because the biofilm, so that matrix itself, can allow bacteria to be more tolerant to antibiotics. So bacteria that would be more easily killed if they were living on their own, when they're kind of protected in that biofilm, the antibiotic might not be able to penetrate, or the bacteria growing more slowly, it might help them to survive. And so then what that will result is like a chronic infection, where you're not able to get rid of those bacteria. So our lab in particular uses this exact bead model to look at how pathogenic bacteria can evolve during infections. And so the kind of questions we think about are, how do bacteria form biofilms? How are they able to communicate within the biofilm and then use this information to disrupt biofilm formation? And then that kind of research can make us think about ways to better treat infections. And there's a little link there if you want to look more at our lab research. Oh, and this was the last discussion question for this part. Just thinking about, can you think of any additional examples of why understanding the importance of these evolutionary concepts is important for solving real world issues?
Don't cancer cells or like tumors evolve like evolutionary when you like assault it with chemotherapy or drugs? So like if you can know like the evolution over time, you can say maybe it's not in our best interest to give this patient drug which will kill all the tumors for right now, but when the cancer cells come back, they are now resistant.
Right, or having those genetic changes. Well, actually, David's probably better at answering the cancer stuff than I am.
No, go ahead. I mean, I think that's a really interesting question, Jonathan. And people are thinking about this in a couple of different ways. Because you're right? So if you add a selective pressure, what grows out are mutations that will be resistant to it. I mean, not exclusively, but you will select for them. So there are this is where thoughts of using like dual therapies come into play. So you're thinking of finding maybe ways that different cells are susceptible and targeting them in different ways. That way you're hopefully killing more cells. But again, we aren't to the point yet where we're able to kill all the cells. So we'll always have this problem of the tumor evolving and growing back and being resistant to therapies. But people are thinking of that. The other way people are thinking of this is trying to drive that evolution. So if we know we give them a drug and they become resistant because of these genetic changes, if we're ready for that, then we're ready with another drug that can take out the evolutionary changes that happen. So we're thinking about being able to push cancers down certain paths of where we know they're susceptible. So that's a really cool question.
Do you want to break into groups just to think about other things quickly, because then we'll have a little time to go over stuff for tomorrow?
Right.
Or if you want, you guys are open, just any other, you can stay in the big group to talk about ideas you could have about thinking about how understanding evolution is important for everything, really.
Well, something that came to my mind was like the coronavirus, and like since it's like an RNA virus, I think I read that like those solely RNA viruses don't have as many like checks to make sure that the DNA isn't mutating. But like we don't want the DNA to mutate and be able to become resistant to any treatments we might have, or not the DNA, the RNA.
Yeah, definitely. I think thinking about coronavirus and how it is evolving is obviously a very active area of research. So they're looking, at mutations that have happened in the virus already. I think as far as my understanding of current research is that none of the mutations that have arisen do they think have made the virus more dangerous or less dangerous. But it's also a good way to track where the virus is coming from. Because if you compare those changes, you can see, maybe in what area a change happened, because a lot of people in that population now have this new viral sequence. And then, people have used it to say, where, let's say in the United States, where did a lot of cases come from? Did they come from China? Did they come from Europe? And I think what they found is they think a lot of that introduction was actually European, but also then being able to just track at a more local level to see how the virus is spreading. So not in a treatment way, but in a tracking way, I think, great.
Any other thoughts? And Amanda, yeah, I think just adding on to what Abby said, I think it is really a heavy area of research, and also thinking of how long maybe the vaccine will work too, if it's evolving and becoming new strains that we will no longer be immune to, like a flu vaccine. Do we have to change it every year? The flu is evolving every year. Will coronavirus, how quickly will it evolve? And again, I'm not a microbiologist or virologist, but my reading of mostly on Twitter and preprints is that it seems like it's going to take a few years, or at least with a lot of coronaviruses, it's taking a few years that we hopefully will be able to be immune once we're immunized. But evolution is absolutely critical to understand how long we will have.
So I think just, are there any more questions on or things people wanted to discuss just about this introduction to the experiments and thinking about applications to different areas of our life? So I'll just move on just to look at briefly what we're going to do for tomorrow. I think this is kind of the coolest part of this week's stuff because it's all the hands-on stuff. So essentially, what you found out was that the changes to these colony morphologies were heritable. And these new mutants actually had higher biofilm production capabilities. And now what we're going to move on to do is to identify what changes occurred in these mutants' DNA that allowed them then to be better biofilm producers. I'm gonna share screen just so I can show you what I posted to the Google Classroom. So here's just so everything for tomorrow is in this little topic, whole genome sequencing and bioinformatic analysis. So you can first just read through what the assignment is, but essentially what you'll do is go through actually 2 slideshows, but one's a bit shorter. And the first one will talk about just walk you through what is the process of sequencing a genome. One way to do it, we're doing it with how we do it is with this Illumina method of sequencing. So that's what that slideshow will review. And then the next slideshow, this bioinformatics one, really is just kind of a step-by-step guide for how to use this online server Patrick to analyze the data that's outputted from the whole genome sequencing process. And so then below that, there's, well, first you can just look at, and we've seen some pictures of this, but here's what the ancestor looked like, typically on the plate. So that's a picture. There's an example mutant that's walked through in that Patrick slideshow. So this is both an image of what did that mutant look like. And then these are the files, they're called FASTQ files that you'll use to analyze the mutant's genome. And that's what all of these other mutant folders hold, are a picture and FASTQ files. And just so everyone so we can look at what all 10 mutants' genomes look like, where the mutations happen. You guys are each assigned to an individual mutant, and your names are just in that little file thing. Does that make sense? And then the assignment, it's described in the assignment overview, the assignment for you guys to do, but really it's just sharing, you analyze this this particular mutant and you figured out that the mutation happened in gene A, and a little bit about what you think that gene might be doing based on your analysis, and a little bit of follow-on where you can look at some information on let's like at the Pseudomonas genome database. So in that in this bioinformatics slideshow, it'll show you how to go there too and find out more information about your gene. So what questions do you guys have about the materials for tomorrow? And if this will be the first time we've done this on a massive scale of people trying it out. So of course, if you have any issues, just message us and we can help you if you're having problems analyzing the data.
Excellent. Well, if there are no other questions, then we'll see you tomorrow. Our first, Salma, what time is Davin speaking? I know you just told me this, and I just posted it in Google Classroom, and I already forget. Oh, you're muted, Solomon.
Sorry about that. Tomorrow at 2.
Okay, so tomorrow at 2, guys, I have someone coming who used to be an admissions officer at Rochester, and then he worked for a company called CollegeWise that essentially people paid him to work with with a student to try to get them into whatever school they want to go to. So he knows a lot about the application process, the Common App admissions process, what admissions officers are looking for.